In this video, I will be introducing the senses. Senses are windows to the world around us. As mammals, we have senses that are centralized partially on our head. So the cephala has many specialized sensory organs such as ears, eyes, noses, tongues, and other sensory organs that can pick up information about the world around us. These senses are going to start a response and send signals to the brain. The senses communicate using the nervous system. The sensory input is going to be turned into an electrical signal that is sent to the brain for integration. The brain will process information from the outside world or our internal bodies and send out signals for a response, such as a motor output to cause us to move. There are many types of receptor cells. We can categorize these based on the mechanism that causes a depolarization. Mechanoreceptors sense physical deformation. For example, changes in pressure can open ion channels and cause a depolarization. Stretching the cells can also cause depolarizations. Chemoreception transmits information about solute concentrations of a solution and picks up different chemicals in the environment. Thermoreceptors respond to heat and cold. We have thermoreceptors both in our skin to sense the outside temperature, as well as internally to sense the internal body temperatures. Pain receptors detect stimuli that reflect harmful conditions. Often these respond to things like excessive heat, pressure, or chemicals that can cause damage to our tissues. Pain receptors are often naked nerve endings embedded in our tissues. There are also many receptors that pick up electromagnetic information. Photoreceptors are one example. These sense light, which are in the visible range of the electromagnetic spectrum. Other electromagnetic receptors are found in animals as well. Some of these pick up electricity, magnetism, or even infrared. All receptor cells work around a depolarization event. Remember, we've talked about depolarization before. This is a rapid change in the charge across the cell's membrane. Each cell has a resting potential that is negative on the inside of the cell and slightly positive on the outside of the cell. When some stimulus occurs, we see a depolarization event where there's a rapid change in the charges. Positive ions will rush into the cell, negative ions may rush out, and this causes the change in the polarization. After a sensory cell is depolarized, it can pass on that information to neurons in the nervous system. Neurons will pass the information on to the brain for integration. The first sense we're going to look at is hearing. Ears respond to sound waves coming from the area around us. Sound is produced by the vibration of molecules. This causes the waves. Volume is a measure of the intensity of these vibrations, and pitch is determined by the frequency of the sound waves. Our ears are designed to pick up these sound waves and funnel them further in to the inner ear where sensory reception actually occurs. The outermost part of our ear as a mammal is called the pinna. This funnels sound through the auditory canal and into the tympanic membrane. The tympanic membrane, also known as the eardrum, vibrates when the sound waves hit it. These vibrations are then transferred to the 3 tiny bones of the middle ear. Vibrations in these 3 bones are then transferred to the oval window. As the oval window vibrates, it transfers these sound waves to a fluid-filled cochlea. The cochlea has fluid that is under pressure. The vibrations cause the basilar membrane to vibrate, and sensory cells inside that membrane are depolarized. This depolarization generates nerve impulses that are transmitted to the brain. Inside the cochlea, we see the hair cells, which are a type of mechanoreceptor. As sound waves move through the cochlea, we see that these hair cells are shifted. The hairs are small protrusions sticking up off the cell body. When the sound waves cause these hairs to move, ion channels are mechanically opened, causing ions to rush in and out of the cell, creating the depolarization. The depolarization is then passed to neurons, which cluster together into the auditory nerve. The auditory nerve runs to the brain. The semicircular canals of the ear are responsible for balance and sensing movement in animals. As in the cochlea, we see hair cells protruding into the fluid-filled semicircular canals. As we move our head in the position of our body, these hair cells will be shifted based on the force of gravity. This will give us information about our orientation and helps us to maintain balance. If you ever have an inner ear infection and you feel dizzy, there's generally problems with these semicircular canals. Our skin is full of different receptors, including some mechanoreceptors. Many of these mechanoreceptors are neurons that have the endings coated in connective tissue. These connective tissue pouches will distend when pressure is placed upon them, causing a depolarization. Thermoreceptors in the skin are sensitive to temperatures in the outside world. Pain receptors are also found in our skin, and these are sensitive to stimuli that could cause damage. We are going to look at two types of chemoreception. We'll look at olfaction, or the sense of smell, and gustation, the sense of taste. These two are closely related. If you think about it, if you're ever in a bakery, for example, and you smell the bread, and it smells so good you can taste it, this is actually because these two senses are so closely related. Whenever we look at aquatic animals, there's actually very little distinction between these two senses, and they are just picking up chemicals in general from the water around them. For our sense of taste, we use our tongues. On our tongues, we have taste buds, each containing 60 to 80 sensory receptor cells. These are chemoreceptors. They're stimulated by chemicals in the food we eat and the things we put in our mouth. Each of these chemical receptors has a slightly different active site to pick up different types of molecules. When those molecules bind to the receptor proteins, it opens a channel, allowing the ions in and causing a depolarization in that receptor cell. We have 5 major groups of taste buds, or taste receptors: sweet, salty, sour, bitter, and umami. These are not entirely in clusters, they're spread out throughout our tongue. Our sense of smell works very similarly to our taste. Chemicals will enter our nasal passage, where those chemical receptors are embedded in our nasal cavity in a wet mucus layer. The chemicals will bind to these receptor cells, opening these receptor proteins, causing a depolarization. It is interesting to note that the chemoreceptors are sticking out through a layer of bone in our noses, and these cells must protrude into a deeper layer to touch the neurons that will then go to our brain. Dogs are amongst the most smell-sensitive vertebrates in the animal kingdom. 5 million receptors are found in the nose of humans, which seems like a lot until you look at a dog who has 220 million receptor cells in their nose. This makes dogs tremendously efficient at finding things like drugs, explosives, and even cancer. Dogs are well known for their sense of smell, but there are those who are even more sensitive. On land, we see that bears are the top smellers, and in the water, sharks are the top chemoreceptors. Sharks can pick up a drop of blood from a half a mile away. Photoreceptors detect photons of light arriving from the sun or other light source, or reflecting off of objects. Our eyes pick up these photons and send information to our brains. We've talked about the evolution of the eye in lecture. We know that photoreception didn't start off with big complex eyes. Planaria, for example, have a very simple eye spot that picks up only light and dark. These eye spots work very similarly to our eyes. We see that there are receptor cells that depolarize when hit by light, They connect to neurons that cluster together into a nerve that heads to the brain. There are other types of eyes in the animal kingdom as well. Arthropods have compound eyes made up of many independent visual units. Each of these has a lens to focus the light onto photoreceptors, and they form a crude image that the insect's brain then has to process. Cephalopods and vertebrates both have a camera-type eye. This is an example of convergent evolution. These eyes have evolved independently. In these eyes, we see that photoreceptors are closely packed on a retina, with an optic nerve in the back of the eye sending information to the brain. In the front of the eye, there's a lens that focuses the light onto the retina. Here's a closer look at a human eye. Let's go over some of the major parts. In the very front of the eye is the cornea. This clear layer allows light to pass through it. It helps to focus the light. The iris, found here, is the colored portion of the eye. When you're facing the outside of an eye, that's where we get our eye color. In the center of the iris, we see the pupil, which is the hole that light passes through. The iris has muscles associated with it that can help change the diameter of the pupil to allow in more or less light. Behind the pupil is a lens. This lens focuses the light on the retina. The retina is this yellow layer all around the back portion of the eye. The retina is where the rods and cones, the actual photoreceptors, are located. In the back of the eye, we see that the neurons cluster into the optic nerve. In that area, there's an optic disc. This is a zone in the retina where there are no photoreceptors due to the grouping of these neurons. The outer white portion of the eye is called the sclera. This is a tough protective portion that keeps our eye from being damaged. Here's a closer look at the retina. Light will be coming in from this direction. That light must pass through a layer of neurons before it hits the photoreceptors. Mammals have two types of photoreceptors, rods and cones. Rods pick up light intensity, and cones pick up color. We have several different types of cones to pick up different wavelengths of color. Once these photoreceptors are activated, a depolarization event occurs, and the information is passed to the neurons in front of them. Those neurons will then send information on to other neurons, which cluster into the optic nerve. There are many other cool senses out there in the animal kingdom. We've mentioned how some fish, like sharks, can detect electrical signals using specialized organs on their noses. The platypus is another animal that can detect electrical currents from its prey. Homing pigeons and some other birds have a substance called magnetite in their head to help them navigate. Magnetite actually senses the magnetic poles of the earth. These animals are able to figure out true north. Pit vipers can sense heat. These animals are not called pit vipers because they live in pits in the ground. They actually have small pits in their face near their noses. These pits contain heat sensors so that they can pick up the heat coming off of their prey. Remember, after sensing all of this information from the world around them and from their internal systems, organisms send that information to the brain for integration, and the brain sends out responses. These responses can involve muscles, where we see contractions, or glands, where we see hormone secretion that can cause other responses in the body.